Hello guys, welcome back. So let's start with the short notes video number 9. The first organism we'll be studying is the Clostridium tetani. So write down Clostridium tetani. It causes tetanus. Okay. Basically it causes spastic paralysis. Paralysis, there is absence of movement. Okay. And spastic, it is another term for spasm, which means contractions. And then you should know one more term which is known as opisthotonus. So if there is too much of contraction, then we term it as opisthotonus. Okay, so you can write this down. Too much contraction is known as opisthotonus. Okay, so spastic paralysis where there is too much of contraction leading to absence of the movement. Now this too much of contraction is also known as opisthotonus. The general property of this Clostridium tetani is that it is gram positive and it is a spore forming bacteria. It is a spore forming bacteria. Okay, and the cultural medium for Clostridium tetani is Kith tarotsi. Now this medium name is very important, so please remember that Kith tarotsi medium. In the question sometimes they mention that we have an organism and we have cultured it on the Kith tarotsi medium, so then you should know that they're talking about the Clostridium tetani. Okay, the main symptom of this Clostridium tetani in the patient which you will see is locked jaw. Okay, and lastly the treatment. So the treatment includes anti-tetanic serum, and there is something special about this, that this anti-tetanic serum can be used as a treatment as well as can be used for prophylaxis, also for prevention. Okay, can also be used as prophylaxis. And specifically for prophylaxis, if we talk about, then we have another way. So for prophylaxis we give tetanus toxoid. So anti-tetanic serum can be used as a prophylaxis also and as a treatment also, whereas tetanus toxoid is only used for the prophylaxis. Okay, the next organism that we'll study about is Yersinia pestis. Let's know some general properties of it. This bacteria, it is gram negative, it is oval shape, and it is 2 microns long. Okay, 2 microns long. It gives you a safety pin appearance. Now why is it so? Let's know that also. Because it is intensively stained at the poles. I'll show you how. Just a second. So for example, this is the organism we're talking about. It is oval in shape, as I said. Okay, now this organism, it is intensively stained at the poles. Now doesn't this give you an appearance of safety pin? Okay, so that's why it shows you the safety pin appearance. And if we are to carry out the test, so which test do we do for Yersinia pestis? We carry out ring precipitation test. So this also comes as a question that if a patient is having Yersinia pestis, then what type of test will you carry out? So it is ring precipitation test is done. Okay, this bacteria is responsible for causing plague. You all might have heard this term or this disease plague once in a while in your life. Okay, and this plague can be of 3 types. So let's divide it, okay, into bubonic plague, into pneumonic plague and septicemic plague. The bubonic plague, it spreads through flies, through flea bites, okay, and it affects the groin area. Now groin is another term for bulbon, okay. Groin means bulbon. So what am I telling you here? It is going to affect the groin area. But what exactly in the groin area? In the groin area, it will affect the inguinal nodes, okay. The inguinal nodes will be affected in the groin area. And to check, or for the diagnostic purpose, what are we going to take as a specimen? So the specimen here, in case of bubonic plague, we take bulbus, okay, and we do the microscopy of this bulbus. So you can write it down as bulbus by microscopy. So how do we check for bubonic plague? We take the bulbus, basically we're taking the swab from the inguinal nodes area, from the groin, and then we do the microscopy. Now the pneumonic plague, it spreads through droplets, okay, and it affects lung. Now if it is affecting lung, that what exactly it is causing, we should know that also in the lung. So in the lung, it will cause you hemorrhagic pneumonia. What it causes? Hemorrhagic means bleeding kind of pneumonia, where there will be bleeding in the lungs, internal bleeding. And the specimen, what kind of specimen we take when there is pneumonia or any lung problems? We take sputum, right? So sputum is taken for the analysis. Now septicemic plague, what happens in this? Actually, when there is a patient who is infected with bubonic plague and is not getting a proper treatment or is not treated at all, then the terminal stage of bubonic plague becomes your septicemic plague. So what is this? This is the terminal event of bubonic plague, okay. And you should know there are no human carriers. And for specimen, what will we take? If it is septicemia, so we take blood to check for the septicemic plague. I hope this Yersinia pestis is clear, that causes plague. There are three types: bubonic, pneumonic, septicemic. You should know which plague or which kind of plague affects what part of the body, and you should know what specimen is collected for a specific type of plague, okay. Next, we'll study about rabies, okay. So rabies is caused by the RNA virus, which belongs to the family rhabdovirus, okay. So the family name for the rabies causing viruses is rhabdovirus, and their RNA virus. Second thing you should know is they contain the helical capsid and they are bullet shaped. So the shape of these viruses are bullet shaped and they contain a capsid which is helical in shape. Now if a patient is having this rabies and we carry out the brain biopsy on such patient, then what will we see? We see negri bodies. What do we see? We see negri bodies in the hippocampus. Now this hippocampus is the area in the brain, okay. So negri bodies will be found in the hippocampus, and for treatment, what can we do? So remember, the treatment for rabies is rabies immunoglobulins. What do we give? Immunoglobulins. And for vaccine or for prophylaxis, we have anti-rabies vaccine. anti-rabies vaccine. Now this vaccine is a live vaccine, you should know. Okay, I hope everything about rabies is clear. Now let's move on with the next organism, which is Leptospira interrogans. The general properties of it, you should know that it is spiral shaped. Okay, you can see in the word Leptospira. So spira is for spiral shaped. Okay, it causes a very common disease, jaundice. So it causes jaundice. How do we check for this organism? We carry out dark field microscopy. Okay, so dark field microscopy is used for this detecting of this organism. What causes this Leptospira interrogans? It is caused by rodents. It is caused by rodents. It can also be caused by domestic animals, but mainly caused by rodents. Okay, otherwise it can be also caused by domestic animals. So whenever in the question you are seeing there is a spiral shaped organism and the patient is suffering from jaundice, then the organism is Leptospira interrogans. And what do we use for diagnostic purpose? We carry out the dark field microscopy. Okay, and what is it caused by? It is caused by the rodents mainly, but can also be caused by domestic animals. I hope this is clear. We'll meet in the next video. If you've got any doubts, then feel free to comment, or you can DM us on the Instagram page. So thank you.